Hi, this is Tina again, and this is part 2 of our Bio 201 Lab 8 tutorial. In part 1, we learned how to use Finch TV and copy our sequence, and now in part 2, we're going to learn how to go to the database and do a BLAST, or use a basic local alignment search tool. So we'll start off by going to yeastgenome.org, and then here we'll hover over the word sequence, and it will give us the option to press BLAST. Then we'll have two different boxes here, one that says query comment, which is optional, just add a name or tag to our sequence. And then here we have type or paste a query sequence, so that's where we're going to go ahead and paste in our sequence. And then we'll press this button right here, run wooBLAST, to actually do our blast. The reason the sequence we just put in is called the query sequence is because it is the sequence we're querying about or the sequence we're asking about. When our BLAST hits result page loads, it's going to look like this. So we have three different hits, right? Three different bars or three different possible matches. You'll notice that one of these is not like the others, right? This nice long purple bar here, and we'll see that that's actually our best hit, and we'll see how to distinguish between different caliber hits in a moment. But definitely part of it you can notice is the length. There's also a color coding system. So red hits would be the best possible hits, and then blue hits are the worst possible hits that would even show up in the database. And if we hover over each of these hits, we get a little bit of information about them. The most relevant piece of information is the chromosome number. So here's chromosome 5, and then two different pieces of chromosome 10. So maybe a good time to brush up on your Roman numerals if you need it. If I scroll on down here, I'll see a list of my significant alignments or significant hits. So only two of my three hits actually made the cut here. And you'll notice that each of them has this e-value or expect value, which you might recall from the background material, is a representation of the likelihood that the sequence in the database that it's matching to is not actually the same as a sequence we pasted in or a query sequence. So in other words, we want that value to be really small, and we'll see here with our top hit, it's 1.4 times 10 to negative 160, so really freaking small, which is exactly what we want. And then this other poor hit, that value is only 0.36, so a relatively large value. This is also tied to the colors, so you'll notice there's the exponent here, right, negative 160. The color here says negative p exponent. Right, so 160 is between 100 and 200, hence the purple color. So again, sort of how the color is indicating to us how good each match is. And then you can go ahead and scroll down and you can see the hits, or if you click on the e-value, it will bring you to the hits. So here's our top hit. Again, it's on chromosome 5. We can see how many of the bases actually match. So out of 745 bases that we put in, 737 of them actually match, so 98%. So again, a really nice match here. And we can see this alignment here. And there's two different sequences here. There's the query and the subject. So remember that the query sequence is the sequence that we pasted in, and the subject sequence is a sequence stored in the database. So of course DNA is double-stranded, but if I know the sequence of one strand, I can pretty easily figure out the sequence with the other strand, especially if I can write a computer program to do it for me automatically, of course. So we have a query, what we pasted in, the subject, what's in the database, so only that one strand. We'll talk a little bit in lab this week. By convention, that subject strand, or the one strand that's in the database, is the Watson strand. As you can imagine, the other strand is called the Crick strand. But we can see the alignment here between our query and our subject, and we have this numbering associated with it. So query 1, that's the very first base that we put in of our query sequence, and we can see our GAATTC, so that EcoR1 site. And then the subject has its own numbering as well that's just based on how the chromosome is lined up and how it's stored in the database. And we'll come back to those numbers later for when we're thinking about drawing our maps. But I can scroll through this alignment. I can see there's lots of nice matches here. So these tick marks represent where the bases are the same, not base pairing. Here's a little bit of a gap here, so why I don't have a 100% match between the two. But you can see overall it's a really nice match. We can also, out of interest, scroll down to the other two. So they're very short and they're not very good alignments. You can see all of these missing matches here. So we're just going to focus on our good hit, and we'll scroll back up here, and you'll see above each hit there's a button for the genome browser, which will take you to this location in the genome browser. So we'll hover over our best hit, and then we'll press genome browser to load it into a new tab, and in part 3 we'll talk about how to actually use the genome browser.